Hello grade 12s, welcome to our channel once again. My name is Veleni Nkosi. In this video we are looking at genetics for grade 12s. So we will be looking at monohybrid crosses. So in monohybrid crosses we will be working on Mendel's principle of segregation. We will also learn about the types of dominance, and then I will also do examples involving genetic crossing. So these are the content for this video. Without wasting more time, let's get to it. So first of all, I would like to start with the types of dominance. So we have three types of dominance, which is complete dominance, incomplete dominance, and the codominance. So complete dominance, one allele is dominant and the other is recessive, such that the effect of the recessive allele is masked by the dominant allele in the heterozygous condition. So here I would like to show you the examples of a complete dominant. We have a yellow seed here which is crossing with a green seed. What they produce yellow seed. That is mean the yellow has masked the green seed here. So this is what we call a complete dominant. And then next up is incomplete dominant. So in incomplete dominant, neither one of the two alleles of the gene is dominant over the other, resulting in an intermediate phenotype in the heterozygous condition. So here neither the allele is a dominant. So example, I have a red flower here which is crossing with a white flower. So what they produce, the offspring is pink flower. So neither of the flower has been expressed at the offspring. So this is incomplete dominant. And then last type of dominant is codominant. In codominant, both alleles of the gene are equally dominant, whereby both alleles express themselves in the phenotype in the heterozygous condition. So example of a codominant is a brown bull and a white cattle. So as they cross, they produce an offspring that the color of the bull is expressed at the offspring. Also the color of the cattle is also expressed. So this is codominant. They equally expressed at the offspring. So this is the codominant. And then next up we go to the crossing. So before we go to crossing, we have to do some terminology that is required. So before we solve a problem involving crossing. So we have phenotype, genotype, homozygous, and heterozygous. So these terms, in our previous video, I explained them thoroughly. I explained them in full. So in this video, I'm not going to go miles trying to explain them. So if you haven't watched the video, then I've put the link above, then you can watch the video. So let me start with phenotype. Phenotype is the physical appearance of an organism. So phenotype is how the organism appears. So like a yellow seed, this is the phenotype. So because the seed appears yellow, so yellow seed, this is the phenotype. Or blue seed, also this is still let me say green seed. So this is still a phenotype. Or a white flower, white flower is still the phenotype. So phenotype is the appearance of an organism. And then genotype is the combination of alleles. So this is where we combine alleles. Let's say a yellow seed and the green seed are crossed, and then we want to see the result. So when we write a genotype, because in our previous slide we say yellow is dominating, so we will write yellow as a capital letter. So yellow, because it's dominating, we write it in a capital letter. And then green, because it's recessive, we write it in a small, we write it Y as a small letter, because green is recessive to yellow. So green is represented by Y, while yellow is represented by Y. Wow, capital letter Y. So we won't write G, we write Y, but we represent green because it's recessive. Like if we say someone who is tall, and then we say T represent tall. So if short is recessive, then short, we're going to write small letter T. Here we say this person or this genotype is tall, for tall, T for tall, and then small letter T for short. This is the recessive, this is dominant. Or we can write T and T, or complete recessive T and T. So this is another example of a genotype. And then homozygous. In homozygous, homozygous is when an individual has two identical copies of an allele. Like if you have a capital letter Y and the capital letter Y. So here you have two identical copies of an allele. This is how we write homozygous. Or we have small letter T and the small letter T. As long as if they are the same, they are identical. Or we can have something, as long as if they are identical, like small letter, big letter R, maybe this represents red. If you have red and red, this condition we call it homozygous. And then heterozygous is when an individual has two different alleles. Like if we have T for tall and then small letter T for short. Here we have two different alleles. This is a heterozygous. Like if we have a capital letter Y and then small letter Y. So still here is still heterozygous, because this Y might represent yellow, but this one is not representing yellow, it's representing another color, maybe green or black. As long as if this one is dominant, this one is recessive. So the small letter represent recessive. So if you haven't watched the video, then go and watch it. I've explained it and make some examples. Then now we go to Mendel's principle of segregation. So Mendel's principle of segregation say organism possess two factors which separate or segregate so that each gamete contain only one of these factors. So here this law tells us any organism has two factors. So like here we have a tall and then short. So or we can have tall and tall, or tall and something. So before fertilization has taken place, these factors must separate, and then each gamete will receive only one fact. Like here, if we have T for the genotype T and then T, so this, it will undergo segregation. So when it undergoes segregation, we'll have T and then T, which are separate. We'll have one gamete and then another gamete. So this is how the factors are separated. This is the process of segregation or meiosis. So this is meiosis. Actually, this process of segregation is meiosis. So these are the things that you need to know before you solve the genetic crossing. So now let's go to example of genetic crossing. So if you are going to solve genetic crossing, you must write it like this one. This is how you will get the marks. So if you write it somehow, you won't get the marks. You must know that this P1 is the parents, and then the phenotype is the phenotype of the parents that are crossing. So if it's yellow, you will write yellow here. If it's yellow and white, you write yellow here, you write white here. And then here is the genotype. You will write the genotype of the parents. So like if it's yellow, obviously it's yellow, and then they will tell you if it's monozygous or heterozygous. If it's heterozygous, then you are going to have yellow, which is dominating, the one that we see, and then the recessive one, you will write it here. And then this side also, you will write the one that is dominating and the one that is recessive. If they are both dominating, then it's okay. They will tell you that this is homozygous. You know that if it's homozygous, then the organism has identical allele. So here, after that, they undergo meiosis. So you write gametes. When you write gametes, you separate these factors here. You will separate them. Then you write them here, one here, and then one here, one here. And then during fertilization, you will write factors of one organism in this row, and then you will write another in this column. Then thereafter, you will apply, you multiply them. This one, you multiply this one, you write it here, and then you write it here, we write it here, we write it here, and then we are going to have, when we multiply, because this is a gamete, and a gamete, a gamete, and a gamete. So here we are going to have the genotype, and then you will write the genotype here. This one, you will put it here. This box will be here, and then this box will also be here, and then this box will also be here, and then after you write the phenotype. So let me do quickly do one example, then you will see what I'm talking about. Here is the example. It says a seed can be round or wrinkled. So here they are telling us it can be round or wrinkled. Use genetic cross to show the genotype and the phenotype of the first generation. F1 is first generation, when 2 heterozygous. Here they are telling us it's heterozygous plant with round seeds are cross. The allele for round seeds is dominant over the allele for wrinkled seeds. Use the R for round seeds and small letter r for wrinkled seed. So here we have all the information. So for example, so I will take out the information, then you will see how I took it out. What they are saying, they say the seed is round or wrinkled. Then they say use the genetic cross to show the genotype and the phenotype. This is the final answer of the first generation. First generation is F1. So they need us to write what we will see when 2 heterozygous. So if they say it's heterozygous, that is mean our genotype doesn't have identical allele, it has different allele. So heterozygous plant with round seeds are cross. So they say they are round because they are both round. And then our phenotype is round. Round seeds multiplied by crossing with round seed. This is the phenotype. It's because it's heterozygous and then with round seeds are cross. They are both round. And then the allele for round seed is dominant over the allele for wrinkled seed. Because they say they are heterozygous, that is mean they said R is dominating the small r. So for it to be round, we say because they are heterozygous, we are going to have R and then R. Also this side, we are going to have capital letter R and the small r, because R is dominating, and then this is important. They say heterozygous. So we are going to have capital letter R, small letter r, and then capital letter R and the small letter r. And then these factors here, they will undergo meiosis. And then when they undergo meiosis, we are going to have gametes. So our gametes, we are going to have R and then R. Even in this organism, we are going to have R and then R. So here is the process of segregation, or is the principle of segregation or meiosis. And then thereafter, what will happen is fertilization. So fertilization, the gametes are fusing. So let me write the gametes for this. Here we have R, then we have R, and then down again is for this one. So we have R and then we have R. Now we combine them. Here it's R, and then R and the R this one. So we have R and R, then capital letter R and R. They will make a R and then R, small letter r and small letter r, which we have R and R. And then we write for F1, this is the offspring. We will write the result inside here. So we have R and R. Again, we have R and R. We have R and R. And then we have small letter r and the small letter r. So this is the result, the genotype. So the phenotype, we have R and R, which is round. Even this one will make round. This one will also make round, but this one will make wrinkle. So for phenotype, we have 3 round, and then we have 1 wrinkle. We have 3 rounds and then 1 wrinkle. This is the answer, because we have R and R, which will make a round, And then R and small r, they say R is dominating, that is mean here we still have round. R and R, here we still have round. So here we have 2 recessive, that is where we will have the wrinkle. So this is the answer. The phenotype is 3 round and the 3 wrinkles. This is how we work with the crossing or genetic crossing. So I have another example here. Another example, it say a homozygous red flower plant cross with a homozygous white flowering plant and produce a plant that have a pink flower. So complete a genetic diagram using a R for red and a W for white. So here I have my table here. For parent, they say homozygous red flower. So if they say it's homozygous, that is mean our genotype, it has the identical allele. So the allele of the genotype, they will be the same. Okay, so here they say homozygous red flower plant cross with homozygous white flower plant and produces plant that have a pink flower. Complete a genetic diagram using R for red and W for white. And then so a parent one is red, or I can say red flower, and then crossing with a white flower, and then they say it's homozygous. So if it's homozygous, so we are going to have red and then red, and then we multiply by a white is for W, multiply it with white W. These are the capital letters W and W. So because both are homozygous, and then they will undergo meiosis or the process of segregation, and then we will have R, then R, this side we have W and then W. So we will write them at the fertilization, they will undergo fertilization, and then we have R and then R, and then we have W and then W. So in this box we will have W and R, and then we have W and R. Also here is W and R. Again here is W and R. So these are the result. So the genotype, we have W and R, W and R, W and R, W and R. This is the genotype. So phenotype, so that is mean W and R, they make pink because they say they will produce a pink flower. So that is mean we have 4 pink flower. So here you have showed that when you cross white homologous flower and then red homologous flower, you end up with a pink flower. So WR, it's pink flower. So this is how you work with the genetic crossing. So the most important thing, you must know how to fill up this. P1 is for parents, and here you write the phenotype of the parents, and then we write the genotype. The genotype, they will undergo meiosis, and then you have the gametes, which is the genotype, will separate them, and then fertilization, you put them in this square, and then I can say you multiply them or add them, and then thereafter you have the F1. F1 is the first filiate generation, or is the first offspring. Then the genotype of the first offspring after fertilization is genotype of the first offspring. Then you write the genotype, then phenotype. This is the phenotype. So I hope I make sense. So if you have watched this video or there is something that you did not understand, please comment below, then tell me what you didn't understand. So if you have watched this video to this far, please give the video a thumbs up and subscribe to our channel. If you are studying, good luck with your studies. God bless you. Thank you very much.